In a groundbreaking research, researchers at the University of Rochester have successfully transferred a longevity gene from naked mole rats to mice, which resulted in improved health and extension of mouse lifespan. This is important because can we actually increase our lifespan too? The principal investigator of this research, Vera Gorbunova, said, "Our study provides a proof of principles that unique longevity mechanisms that involved in a long-lived mammalian species can be exported to improve lifespans of other mammals, which include humans."
Human lifespan can be extended. We can probably live a lot longer with the help of science. But what's very important is to live healthy. If we could live up to 200, 300 and stay healthy and productive, that would be fantastic, and that's what we hope to achieve, or at least get closer to this goal through research.
First of all, why naked mole rat? Naked mole rats are exceptionally long-lived species with a maximum lifespan of up to 41 years, an 8 to 10 times longer lifespan than similar-sized mice. Second, unlike many other species, naked mole rats do not often contract disease, which include cardiovascular disease, arthritis, and cancer when they age. That brings us to next question. What is specific factor of these naked mole rat are causing them to live longer than comparable rats? These same researchers previously discovered that high molecular mass hyaluronic acid is one mechanism responsible for naked mole rats for their resistance to cancer. In fact, compared to mouse and human, naked mole rats have about 10 times more high molecular mass hyaluronic acid. In their previous study, when they removed HMM-HA from naked mole rat cells, the cells are more likely to form tumors. So, in this study, these researchers wanted to see if the positive effects of HMM-HA could be transferred to other mammals. So, to accomplish that, this team genetically modified a mouse model to produce naked mole rat version of the hyaluronic acid synthase 2 gene. This gene is responsible for making a protein that produces HMM-HA. This team designed a mouse model which overexpressed naked mole rat has2 gene. They call it NMR has2 mice. NMR has2 mice showed lower spontaneous cancer incidence compared to their control group. Also, this difference is more amplified for the oldest age group. For mice older than 27 months, cancer incidence was 83% for control group and 49% for NMR has2 mice. Next, NMR has2 mice showed an increase of 4.4% in median lifespan and 12.2% in maximum lifespan. Also, to quantify improved health span in this mice group, these researchers used a mouse frailty index, which include a lot of parameters that include body weight, temperature, rib strength, mobility, vision, and hearing. Although the frailty index score increased with age for both MMR has2 and control mice, however, the frailty index score of old MMR has2 mice was substantially lower than that of the age-matched control group. Another finding of this research is that they found that in the MMR has2 mice, they had reduced inflammation compared to control mice. Most importantly, this research also presented a model how HMM-HA work for longevity. First of all, HMM-HA produced by overexpression of the NMR has2 gene protects tissues from oxidative stress. Second, it improves maintenance of intestinal stem cells to provide a better gut barrier function during aging, and finally reduces the production of pro-inflammatory molecules by immune cells. So the takeaway from this study is that by overexpressing a gene that produces HMM-HA, you can extend lifespan and improve health quality of mice. Now, how can we apply this finding or this research for human life? This research is important because it gives us a possibility of how HMM-HA could also be used to improve human lifespan and probably to reduce inflammation and other disease like heart condition. So how come this research benefit humans? Dr. Gorbunova said, it took us 10 years from the discovery of HMM-HA in the naked mole rat to showing that HMM-HA improves health in mice. Our next goal is to transfer this benefit to humans. So they believe we can accomplish this in two methods. First of all, we can either slow down degradation of HMM-HA because as we age, our body tends to degrade HMM-HA. So if we can slow it down, that could be one way we can accomplish to achieve this goal. And the second way, which more aligned to this research, is that can we enhance HMM-HA synthesis? Dr. Silionov said, we already have identified molecules that slow down hyaluronic acid degradation and are testing them in preclinical trials.